It is the anti-cancer and other pharmacological prospect of the
Aspects
green
of green nano.
particle derived from selected plants.
Yes, sir.
Okay, so this is your synopsis, or you are going for some? You are going for RDC.
Yes, sir. Tomorrow.
Tomorrow will be your RDC.
Yes, sir. This
Okay,
is what I have prepared the PPT for the synopsis as well as for the PPT
okay. This you prepared
presentation.
yourself.
Yeah, I have prepared by myself, sir.
Okay, your supervisor name
I
is what?
have Meenu ma'am.
Minu Sharma.
Yeah,
Okay.
Meenu Sharma ma'am.
Synopsis of the synopsis, then a space is required, then of the thesis.
Synopsis of the thesis. Oh, okay,
There
okay,
is a
sir.
space.
Yeah,
Okay.
yes,
Or
sir.
not required to write of the thesis, only synopsis is sufficient. Synopsis submitted by Rency George, enrollment number, a
Okay.
partial fulfillment of the requirement for the degree of enter Doctor of Philosophy in subject zoology. In
Yes,
zoology,
sir.
subject लिखने की there is no necessity to write subject. It is
Okay,
in
okay.
zoology.
Okay, sir. Yeah, sir.
Under the guidance of Dr. Meenu Sharma, Professor, Department of Zoology, Faculty of Science, Mansarovar Global University, Mansarovar Global University, Switzerland. So, what was your motivation to pick this title? How you reached to identify this particular thing that where you want to work?
Sir, because nowadays this medicine new novelty is there, and green nanoparticles are being synthesized from the many years or centuries onwards. They are using the herbal medicines. So I thought of taking the extract and synthesizing the nanoparticles from there, from the selected medicinal plants. I have selected 3 medicinal plants that Tinospora, Andrographis, and Scindilla asiatica. These are the 3 plants that which I have selected.
Why
But
you have selected
main
these three plants? Why not others?
because rest all extensively they have studied. First, I was thinking to take ginger, curcumin, and something else. But then I have seen that in the literature review that is over extensively studied subject. So I thought of going for a this plants.
Okay, the
Already
plants
these
you are
plants they have.
mentioning. So, what is the production of these plant as compared to other plants in India or the particular area where you belong? Availability of these plant.
Yeah, availability of these plants. Yeah, these plants are available, sir, and it is I have read the reviews also for this of these plants. It has anti-cancer properties, anti-inflammation, antioxidative. So due to that only I have selected these plants. So I thought of making it into a
So, do you have
finding a
find
new.
any other literature on these plants? Have there any literature already available?
Yes, sir. Many literatures are available. Then I thought that I will go to the cancer cell lines.
Okay. So, which particular cancers you are targeting with these?
Sir, I also do not have that much idea about it, sir, but I am planning for the cell cultures are being done in the biotech labs, no? So I'll be taking one of the cell culture and I will be doing it. I will discuss with the guide and I will be planning it.
Okay,
Right
move
now, I
ahead.
don't.
Okay, move
Yes,
ahead.
sir.
Move ahead with the next slide. Yeah, explain here.
Introduction. Shall I?
Yeah, you start. You
Yeah.
continue.
So, cancer, since it is a global health concern and leading cause of death worldwide, accounting for millions of deaths annually, the conventional therapies such as radiotherapy, chemotherapy, surgeries, I meant by that only conventional therapies, often have limited efficacy and severe side effects. So, we need to need for a novel anti-cancer therapies. So it mainly target on cancer cells, specifically with minimal damage to the healthy cells. overcome resistance to conventional therapies. And nowadays, it's nanotechnology, role of nanotechnology is emerging, and nanoparticles can target cancer cells and deliver the therapeutic agents. Green particles, that is GNPs, synthesized from plant extracts, offer a promising approach. It can investigate cancer and other from the selected plants such as Tinosporia cordifolia, Andrographis paniculata, and Centella asiatica. Green synthesis of these nanoparticles for cancer treatment, it's a major health concern worldwide. So conventional treatments have limitations and side effects. The natural products and green technology offer the promising alternatives. Role of natural products in cancer treatment. Medicinal plants have been used for centuries to treat diseases, various types of diseases like diabetes, arthritis, such as different types of various types of diseases. And also in a special way, cancer also. Plant extracts have been used to synthesize nanoparticles with enhanced bioactivity and biocompatibility. Then importance of green synthesis. Green synthesis is preferred method for synthesizing the nanoparticles, as I have mentioned earlier. Natural products are used as reductants to reduce the metal ions to nanoparticles. This approach is eco-friendly, cost-effective, and produces biocompatible nanoparticles. Potential of silver nanoparticles in cancer treatment. These silver nanoparticles have shown antimicrobial, wound healing, and anti-cancer activities. Green synthesis of silver nitrate, that means AGNPs, using natural products is a promising approach for cancer treatment. So I would like to conclude the introduction here. So green synthesis of nanoparticles using natural products offers a promising approach for cancer treatment. Further research is needed to explore the potential of this approach and to develop the effective and safe treatments for the cancer. Main objectives of my studies: to explore the anti-cancer potential of green nanoparticles derived from Tinospora cordifolia, Andrographis paniculata, and Centella asiatica. Second objective of my study is to investigate the pharmacological properties of green nanoparticles derived from Tinospora cordifolia, Andrographis paniculata, and Centella asiatica. Third objective of my study is to evaluate the potential of green nanoparticles derived from these three plants, adjunct therapy for cancer treatment. Then fourth one is to examine the role of green nanoparticles derived from Tinospora, Andrographis, and Centella in modulating the key signaling pathways involved in cancer. Fifth point is to identify the challenges and future directions for the development of green nanoparticles derived from these plants as anti-cancer agents. Methodology used here is plant material collection and authentication. The selected plants are these three: Tinospora, Andrographis, and Centella. It will be collected from the local markets and authenticated by a botanist to ensure their identity and purity. The plant materials will be carefully selected, cleaned, and dried to remove any impurities or moisture. The dried plant materials will then be powdered using a grinder and stored in airtight containers to preserve their bioactive compounds. The authentication process will involve morphological and microscopic examination of the plant materials, as well as verification of their botanical names using standard reference text and online databases. The extraction of plant extracts will be done in this process. The dried plant powders will be extracted using a solvent, ethanol or methanol, to obtain the plant extracts. The extracts will be filtered, concentrated, and stored in the airtight containers. The extraction process will be carried out using a Soxhlet apparatus, which allows for the efficient extraction of the bioactive compounds from the plant materials. The solvent will be carefully selected based on its ability to extract the desired bioactive compounds and extraction conditions, such as temperature, time, solvent to sample ratio, etc. will be optimized to maximize the yield and quality of the extracts. The extracts will be then filtered using a Buchner funnel and Wortmann filter paper to remove any insoluble residues. The filtered extracts will then be concentrated using a rotary evaporator to remove the solvent and obtain a semi-solid extract. The concentrated extracts will be stored in airtight containers to prevent degradation and contamination. The extracts will be labeled and stored in a cool, dry place until further analysis and use in the synthesis of green nanoparticles. So statistical analysis will be done. The data obtained from the various assays will be analyzed statistically using ANOVA and Tukey's test to determine the significance of the results. The results will be presented as mean SEM and P lesser than 0.05 will be the considered significant. The data obtained from the study will be analyzed statistically using various parametric and non-parametric tests to determine the significance of the differences between the treated and control groups. The statistical analysis will be performed using statistical software packages such as SPSS or GraphPad Prism. The normality of the data will be tested using the Shapiro will test, and the homogeneity of variance will be tested using the Levin's test. The comparison of means being the treated and control groups will be performed using the one-way analysis of variance or the Kruskal values test, depending on the normality and homogeneity of the data. The post hoc analysis will be performed using the Tukey's test or the Dunn's test to determine the significance of the differences between the groups. The correlation between the variables will be analyzed using the
Yes, you continue.
Pearson's correlation coefficient or the Spearman's rank correlation coefficient. The results of the study will be presented as mean standard error of the mean SEM for median interquartile range depending on the normality of the data. The p-values will be calculated and the results will be considered statistically significantly if p lesser than 0.05. The statistical analysis will provide valuable insights into anti-cancer, anti-inflammatory, antimicrobial and antioxidant activities of green nanoparticles and will help to establish their potential therapeutic applications. Then synthesis of the green nanoparticles, plant extracts used as reducing agents to synthesize GNPs. Then metal ions will be used, silver or gold will be used, reduced using plant extracts. Reaction mixtures stirred, heated and monitored using UV vis spectroscopy. Then characterization of GNPs, techniques used is TEM, SEM, XRD, FTIR, UV vis spectroscopy, DLS. Then advantages of green synthesis is that it's eco-friendly and cost-effective. It eliminates the toxic chemicals, scalable and biocompatible. Then applications of green nanoparticles, anti-cancer activity, anti-inflammatory activity is there, then antimicrobial, and it is good for biomedical applications. Then a review of literature is being done. Review of literature. Shall I continue with review of literature? So according to Murad Hashim et al., cancer remains a significant threat to human health, necessitating the multifaceted approach to intervene in its complex signaling pathways. Plants offer a rich source of phytotherapeutics with promising anti-cancer properties, supported by epidemiological data linking vegetable and fruit-rich diets to reduce cancer risk. This review highlights the anti-cancer activities of key phytochemicals, including their antioxidant, anti-inflammatory and molecular modulating effects,
So your
as well as... Pardon?
Methodology.
Methodology, yes, sir.
Rajneet, methodology.
Yes, sir. Methodology. Plant material collection and authentication. That was my methodology. I have
We
already.
just share one PPT that's we prepare as prescribed by the university to Rency George, so that she just rearrange her work. Principal, basically, whatever your work is, no, that is not properly arranged. First thing, you have to be properly arrange your work under specific heads and properly give a tablet edge. Like your literature survey, that should be a tabulated form. Okay, and
Okay,
you
sir.
have to identify the base paper on which you are doing your research or extension of your research. As you suggest that there are lots of research already carried out on ginger or even the turmeric and other, while there are very few research you available on the paper plant
These three plants.
you are taking for your research.
Yeah.
So for the base of that, you must have a one or two paper that those who are doing the work on this particular field and identified that their extract and particularly nanoparticle of those plant extracts are beneficiary for various medical uses, including the cancer and anti-inflammatory and other things. Okay, so what the those researchers are doing currently, that will be reflected from that literature survey. Okay, that will be termed as the and what extension to their work you are going to do. Now, one another thing, like you are mentioning the lots of machinery like XRD and others. So, you also mentioned that these materials should be, or these instruments or machines should be available. Definitely must be available with your university or with you. To first check out
Yeah,
the.
that I have to discuss with the guide, sir. I have not discussed so far it. I have just submitted my synopsis. At the same time, I have sent this PPT also to her.
Okay, so just first of all, just rearrange your PPT. What I am finding that there are lots of material you are providing in your PPT that is not exactly required. Okay,
Yes, sir.
we have to focus on what exact proposal we are putting in front of the panel on which you are going to research. Okay,
Yeah. Yes, sir.
first of all thing, after just giving a brief introduction, you have to explain the what is the scope of this research, what are the significance of this research that you have to explain. After getting that scope and significance of research, what is your objective to doing this as you identified from the research graph from the base papers. Okay,
Hmm. Yeah.
that you have to be narrate to them that I had gone through lots of literature survey and
Hmm.
identify that research gap. The people there are lots of research available of these plants, but still they are
Hmm.
not conclusive. Okay,
Hmm.
even though
Hmm.
there are few more plants which have the almost similar properties and maybe proven more a stronger competitor to others anti-cancers agents and activities, so we can go with them also. So I want to research on these paper. Based on these 4 or 5 base papers, you have to list the name of those person or people who are doing the research on your line of action. Okay, after
Okay,
that,
sir.
on the basis of basis of the previous research as carried out by the various researchers, first what I have to do as per my research methodology, one thing that identify after identify the plant, I have to uh, what I say focus on availability of the materials from where I
Yeah.
can find out these plants. What is the availability in terms of
Hmm.
geographical
Hmm. Yeah.
region or geographical positions of my city or place or university, whatsoever you want to consider.
Hmm. Hmm.
Then you have to be capitalized about the process, how to extract that you had explained that what exactly you are going to you will be do with your material and do the extraction process, and what type of machinery, not only just the mixing of ethanol methanol for getting the nanoparticle, you require a machinery for that. what type of machines will be that, and how you will proceed the extraction, what will be the quantity, how you prepare the sample for the XRT or GAM test, etc. What will the process of preparing a specimen, how you will how many specimen you require to do that. And that is the just preparation and method of the preparation of your medicine or the nanoparticle, and the testing of the nanoparticle. And next thing is their trial, on the clinical trial, or we can say in the trial in terms of your lab, using the cancerous cell and the treating those cancerous cell, or look out the activity of cancer cell under the microscope. That will be your next word, how you will proceed with that, what type of specimen you are preparing from, what will be the source to getting all those cancer cells, and how you are treating all those with you, then what are the significant changes you had observed in the due course of period. And then actually your conclusion ki this is my whole journey that I want to prepare. And at the end, there may be a question, are you going for the clinical trial, human trial, or animal trial? So there may be a question ki no, I'm not going for the clinical or animal trial and all. So that we can put all together ki how we are doing that. We can say it's a future scope that people can go with that. Okay, so by this way, you have to properly arrange your whole thing. Now, first, Rajni just share one PowerPoint presentation of synopsis of Mansarovar University that you have previously had, so that Rency ma'am may correct his formatting of his her PPT. Okay. Basically, there need a strict format is provided by the Mansarovar Global University that you have to follow. Okay.
They have sent the format. Based on that only I have prepared, sir. They have told me to make based on your synopsis. So based on the synopsis, I have gone. I have made
You
it.
just seen whatever the presentation Rajneet shared with you, and what the changes that you required. Just adapt all those things in your PPT. Basically, formatting of your PowerPoint presentation is the important thing. You didn't format anything. You are just continue copy and paste from your synopsis to your PowerPoint presentation. There must be a follow up of particular formatting in terms of style and justification, center line, size and all font size and other things that you have to be keep in mind.
Okay, okay, sir.
Okay,
Okay, sir.
title of slides and all other things. So
Yes,
Rajni
sir.
will provide you a sample. You just go through that. That
Yes,
will
sir.
help you out and make you more confident in front of your exam.
Okay, sir.
Okay, but just explain them about the base paper or what the present research is going on in your field, and
Yeah.
how differ your researches from them. That will be a term as the research gap that you have to be showcased to them.
Okay.
Okay,
Okay, sir. Okay.
okay,
Yeah.
okay. Rest is okay. Be prepared for tomorrow. All the very best.
Thank you, sir.
Most welcome.
Okay. Thank you, sir.